we get experimental science. We're curious, non-judgmental.
There be a deep dive on pitcher plants. And so we're going to jump into what pitcher plants are and some of the interesting science that's being done on them. But I wanted to start off with a series of images of what exactly these look like. These are carnivorous plants. Do they smell? So the odors, there are volatiles that come out from these. And part of these, the volatiles, is that from that digestive zone. So there is a series of cells. We're going to take some really, really zoomed in looks and seeing what the cells like in here. But those release digestive compounds to break down whatever ends up in there. That does give off odorants as well. And in fact, you can see here, not on this panel, we have a different paper actually where they analyzed the microbiome and the volatiles within that sample and examine what's coming out of it. And no, it doesn't smell like rotting bugs. So interestingly enough, these pitcher plants eat very different things. So there's lots of different species. You have ones that eat a mammal. So there's ones that eat mice. Yes, they actually eat small rodents. There's others that eat insects. There's others that eat bacteria. And so you get a wide range of odor. And actually, there's not really a strong sense of rot. So if they had rot in them, that would actually be negative for the plant. And the reason for that is because they would be trying to digest something that is undergoing necrosis. Or not programmed cell death, but rather just universal, just death of everything. And that breakdown, you can't really get nutrients out of it, and it actually becomes detrimental for the plants or any animal. So in fact, yeah, exactly, it would make them sick. So like some of the ant species that we study that are hunters, they actually sting their prey with a paralytic and eat them while they're alive. But if the cricket dies or spider or whatever they're eating, if those die, they actually get rid of it. Because as soon as something dies, it starts undergoing that necrosis. And that is, even if you did eat it, you're not getting any nutrition out of it. Because the proteins, even the amino acids, everything is just breaking down to the point where you can't utilize any energy from that. Do they have this special different look under UV like other plants? They do. It's different on different species. So Neuralicia brings up a cool point of what do plants look like with insect vision. And so here's a nice example. So these are Rudbeckia. I thought these were Susans. We have these in the backyard. Two images. On the left is what we see as humans, and on the right is what the insect sees. And you can see with the insect vision, there's almost a landing pad of what they're going towards. And so some of the pitcher plants do look different under UV light, but it depends on their goal. It depends on what animal they're trying to lure in. So there's actually some that lure in insects not actually to eat. There's a whole species that tries to get ants to come into it and take out like partially digested things, and that's actually how they avoid necrotic tissue in their the plant itself. So the question was, how close related are they? It does seem like they are independently evolving for carnivorous plants. So these are just pitcher plants that they analyzed in this paper and did the genetic sequencing and found that there were sequence differences associated with becoming carnivorous. But that component seemed to be independently evolving from other carnivorous plants like the Venus flytrap, and the other one that we're going to talk about today is the sundew. So you might predict like maybe those all had a common ancestor and they diverged from other plants and these became carnivorous and the other plants were just photosynthetic. It turns out, based on the genomic sequencing, what it looks more like is that these plants independently underwent gene loss and mutation to become carnivorous. Rather than having it all happen at one time, it happened in 3 independent lineages. So while they are related, they're not as closely related as you might predict, just based off their structure and how they might look different. And some of the changes that occurred in them is they did leaf transcriptions. They found genes associated with prey attraction, capture, digestion, and nutrient absorption. Those are the primary groups of genes that were different on this front versus what you ended up seeing in other organisms. So like independent, if you compared the pitcher plant, the Venus flytrap, and the sundew to other plants, I mean, it all kind of falls under the guise of nutritional changes, but a lot of it's also digestion, just the acquisition of these phenomena. Yes, so Venus flytraps, if you do not give them food, like flies, they'll actually revert to photosynthesis. they'll actually have those little, the bitey bit fall off, and they can survive on photosynthesis. But there doesn't seem to be ones that do simultaneously. And I think that's the hypothesis that I've read on that is most likely because you're using two different modes of energy absorption and energy acquisition, and it's just difficult to maintain that. And so you'd have to do, it's just like one or the other because they use different processes, and the amount of energy expenditure you do to do both at the same time just wouldn't make sense. Instead, you'd either do one or the other, whichever one was giving you more energy. Let's see. They'd actually looked at a fair number of them. These are all different kinds of pitcher plants, and they were looking at different sequences on them and doing that comparative analysis and finding, there's all of these proteins that they're identifying are ones that are also involved in like breakdown of food, nutrient absorption, and actually not necessarily loss of photosynthesis, but rather you are getting some gene loss, so they're just not as efficient at photosynthesis anymore. They've identified a different kind of opportunity to acquire nutrients. I think that they're common in rainforest flora, which doesn't get a lot of sunlight. So not every location, but there are a fair number where it's like the competition for the resource of sunlight might be low, and so they can go for something different. Now that's not the case with the Venus flytrap, which is why the Venus flytrap for that species is much easier to go without having the sunlight, right, or the food, like the carnivorous element to it. You can survive with one if it doesn't have the other, which is kind of neat to be able to see. And then here are some of the overlapping elements that they've identified between different species, looking at digestive fluids and close relatives. So they found, there are some unique digestive fluids in these plants that have evolved that, do what they do to break down the host. But they also have some commonalities with distantly related species, including like bacteria as well, for like digestive fluids and breakdown of nutrients and nutrient absorption, like phagocytic bacteria that eat other organisms. There is some overlap there, which I think is really cool. So that is just one element of how these animals are different.